All right, everyone, welcome. Today, we're going to totally master a classic biology experiment. You know, the one that's a huge favorite for board exams? Yep, we're talking about Mohl's half-leaf experiment. By the time we're done here, you'll know exactly how it works, what it proves, and most importantly, how to crush any exam question they throw at you.
So, here's our game plan. We're going to walk through this step by step, building up your understanding from the ground up so you're totally prepped for your exams. Ready? Let's get started.
Okay, so let's start with the big picture. We all know that plants do this amazing thing called photosynthesis, right? They make their own food, starch, using sunlight. And we've learned the recipe: carbon dioxide, water, and light. But, you know, in science, just knowing isn't enough. You have to be able to prove it.
And that's the real trick, isn't it? For any good, fair experiment, you have to isolate just one thing, one variable. So the question is, how can we set something up to prove that it's the lack of carbon dioxide and nothing else that stops photosynthesis in its tracks? Well, that's exactly where Mohl's brilliant experiment comes in.
Before we can even touch the experiment itself, we got to get our plant ready. Now, this first step is absolutely critical, and it's something students forget all the time in exams, so paying attention right now could seriously snag you some easy marks.
What we're doing here is basically creating a blank slate. By sticking the plant in the dark for a couple of days, it has no sunlight, so it can't make new food. It's forced to use up all of its stored energy, all that starch. This whole process is called destarching, and it gets the leaves perfectly prepped for our test.
Seriously, think about this for a second. If there's already starch in the leaf before we even start, how on earth could we know if any new starch we find later was made during our experiment? We couldn't. It would mess up the whole result. So destarching makes sure that any starch we find at the end, well, we know for a fact it was made under our test conditions.
Okay, our plant is destarched and ready to go. Now, let's build the setup. We're going to go through this piece by piece, just like you would if you were drawing this for your exam.
First up, we just need a simple, wide-mouthed bottle. The wide mouth makes it easy to fit the leaf in. And it's got to be transparent, right? Because sunlight has to be able to get through to the leaf inside.
Okay, step 2. We add our secret weapon to the bottle, potassium hydroxide, or as you'll see it written, KOH. You can use pellets or a solution. Either works.
So, what does this stuff actually do? Think of KOH as a CO2 magnet. Its whole job is to suck up all the carbon dioxide from the air inside that bottle. It essentially creates a little carbon dioxide-free zone. This is a huge point to remember for those one-mark questions.
All right, now this is where the genius of the experiment really shines. You take a cork, split it down the middle, and gently slide a single leaf from our destarched plant right through it. So half the leaf is now inside the bottle, in that CO2-free air, and the other half is outside, breathing in the normal air with plenty of CO2. And that seal has to be airtight, no leaks allowed.
And for the final step of the setup, we just let nature do its thing. We put the whole apparatus out in the bright sun for a few hours. This makes sure both halves of the leaf get the exact same amount of light and water. So now the only difference, the one single variable, between the two halves is carbon dioxide.
After a few hours in the sun, it is the moment of truth. Time to see what happened. We'll carefully take the leaf out and test the whole thing for starch using the good old iodine test.
And wow, look at this. The results are crystal clear. The part of the leaf that was outside, the one that had CO2, it turns a dark bluish-black color. That's a positive test for starch. But the half that was inside the bottle with no CO2, it stays that pale yellow-brownish color. No starch was made.
And there you have it. It's an open and shut case. Because everything else was the same. Same leaf, same sunlight, same water. The only possible reason for the difference is the carbon dioxide. It proves, without a shadow of a doubt, that CO2 is absolutely essential for photosynthesis.
Okay, so you get the science. That's awesome. But how do we turn that knowledge into actual marks on your exam paper? Let's talk strategy.
All right, lean in for this one because this is super important. These are the classic traps that students fall into year after year. Things like forgetting to even mention destarching or not explaining what the KOH actually does. Each of these points is like a check mark the examiner is looking for. Avoid these and you're golden.
So why is it called the half-leaf experiment? Well, it's because the design is just so clever. The same single leaf is used for both the experiment and the control. The part inside the bottle, where we took away the CO2, that's our experiment. And the part outside, in normal conditions, that's our control group. It's an incredibly neat and efficient way to prove a point.
Okay, time for a little pop quiz. I'm going to show you a few real exam-style questions. I want you to pause and think about how you'd answer, and then we'll walk through the perfect response together. Let's do this.
First up, a classic one marker. What does the KOH do? Simple and to the point. Potassium hydroxide is used to absorb all the carbon dioxide from the air inside the bottle. That's it. Full mark.
Now for 2 marks. To get full credit here, you've got to hit 2 key points. First, you say the plant is kept in the dark to destarch the leaves. That's 1 mark. Second, you explain that this is to make sure any starch you find later was definitely made during the experiment. Boom, 2 marks.
Okay, a 3 marker. The trick here is to split your answer. First, the observation. The part of the leaf outside the bottle turns blue-black, while the part inside does not change color. Then, the conclusion. This shows that starch was only made where carbon dioxide was present, proving CO2 is necessary for photosynthesis.
All right, the big one. For 5 whole marks, you have to bring out the whole shebang. You need to describe everything, from destarching the plant to setting up the bottle with KOH, putting it in the sun, and finally doing the iodine test. And, this is crucial, you have to back it all up with a neat, clean, fully labeled diagram. Use a ruler, make your labels clear, and show that airtight cork.
Okay, lightning round. Let's do a super fast recap to lock this all in your brain. What's the aim? To prove CO2 is needed for photosynthesis. What's the first step? Destarch the plant. The key chemical? KOH, the CO2 absorber. The setup? Half in, half out. The result? The outside half turns blue-black. The conclusion? Simple. No CO2, no photosynthesis. Got it? Good.
And that is a wrap. You have officially gone through Mohl's half-leaf experiment from top to bottom. But here's a little something to think about. If we can remove CO2 with a chemical, how would you design an experiment to prove that light is necessary? Ponder that one. Keep being curious and keep exploring. You did great today.